Okay, hi everybody, my name is Ankita and today I'm going to talk about nanoparticle toxicity, specifically silicon dioxide. So nanoparticles are materials that are less than 100 nanometers in every single dimension, and because of their very small size, they have a very high surface area to volume ratio, which makes them very beneficial. They can be emitted through multiple sources and they're highly mobile, so they are fated to enter aquatic and terrestrial systems as they can move very easily throughout our environment. They are very highly used in commercial sources. So they are used in the biomedical industry, pharmaceutical industry, even the energy industry, and you can find them all around us. So they are highly ubiquitous. Just as a quick size scale, a single hair is typically 9 millimeters thick. A typical red blood cell is about 7 micrometers, and then a typical gold nanoparticle is around 50 nanometers thick. So you need about 20 gold nanoparticles to make up the width of a single human hair. And my specific study focuses on silicon dioxide. It's commonly known as silica, and its chemical structure is SiO2. Silicon nanoparticles are around 50 to 5 nanometers thick, and they are non-porous. I use silicon dioxide because it's highly prevalent in human life. It's commonly used as a food additive. It improves the taste quality of certain food items, and it is an anti-caking agent as well. So anytime you open a food packet and you see a little white little packet in there, it says silica, do not consume. That's what basically my study is based on. Silicon particles are specifically very highly mobile and they're easily dissolvable in water, so they typically end up in aquatic systems. And because of this, they can be easily taken up by microbial forms of life, and then they can biomagnify and bioaccumulate into higher forms in apex predators, namely humans. The literature that I read cited that humans can typically ingest about 35 milligrams of silica every single day, which was extremely shocking to me. And supportive literature that I read for my thesis told me that the main way that silica impacts cytotoxicity is through morphological cell shrinkage. It can cause apoptosis in cells. It can cause cell walls to degrade, which allows chemicals to pass through the barrier unregulated. And then it also increases reactive oxygen species levels. So the main method of silicon dioxide cytotoxicity is presumed to be through the increase of oxidative stress, which is done through the formation of reactive oxygen species. And I'm going to dive into all of that in just a second. But my particular study aims to understand the effects of increasing silicon dioxide on Camelina sativa, which is a model organism. And I use Camelina sativa because it's very versatile. It's also very prevalent in everyday human life. It can be used as an oil seed. It can be used as feedstock. It's a source of essential fatty acids for many humans and animals, and it also can be used as a jet fuel. So it's very versatile. It has a highly conserved genomic structure, and it mimics those of wheat and cotton. And it can grow very fast. It doesn't need a lot of nutrients, doesn't need a lot of water, and it's very resistant to common environmental pathogens. So that makes it very suitable to be grown in a lab setting. The plant's genome has also been mapped in its entirety, so any studies done, such as mine, could be used as preliminary data for further studies working on the genomic structure of Camelina sativa. There were 3 main parts to my study. The first part was setting it up, planting the seeds and setting up my plants. The second part was the morphological data collection and the analysis, and then the third part was the biochemical testing. So the first part was the seed and the plate prepping. I had 5 experimental groups for my project, and there were increasing concentrations, and this included a control group as well. I had 5 biological replicates of every single experimental group, and then 25 seeds for every single plate that were planted in a 5x5 matrix. So as a result, I had 625 seeds that I planted for every single trial that I conducted of the study. That gave me a very large sample size and allowed me to account for any outliers or any biases that I may have in my data. The seeds were sterilized using ethanol in a bleach mixture and then distilled with water. And then my agar was sterilized using an autoclave, and then all of my instruments were autoclaved as well. All of my experiments were done under a fume hood to also account for any contamination that may take place. And then the seeds were grown for 2 weeks under consistent lighting and temperature conditions. Right over there, you can see one of the trials that I had set up. It's kind of hard to see up there in the screen, but you can see the control. The agar was a little bit more clear, and as the silicon dioxide increased, the agar got more and more white because of the silicon that was present there. The morphological characteristics that I collected for were the number of leaves, the longest leaf length, the shoot weight, the number of roots, the longest root length, and the root weight, and then also the total plant length and the total plant weight. I used 2 statistical tests to do my analysis: the ANOVA single factor, which tests for significance across all of the experimental groups, and then the t-test, which tests for significance just between 2 specific groups that I could choose. Here are the results from my ANOVA test, and these are listed in order of decreasing significance. This was done using a 95% confidence interval. So a p-value of less than 0.05 would mean that my results were significant. Shoe weight and shoe length were the two characteristics where I saw the highest significant difference between my groups. So comparing the control to the highest group, there was a large amount of difference. Between the root weight and the root length, there was not so much of a difference, and I could not conclude that they were significant. And then here is an example of what a t-test table could look like. So these are the ones regarding the shoe weight, the shoe length, and the number of shoes, total shoes. All of the boxes that are highlighted green are the ones where I saw a significant difference in the results. So for example, the shoe weight, I didn't see any significant difference between my control group and the 0.001, but I did see a significant difference between the 0 and the 0.005, or 0.05, I'm sorry. So all across the shoes, I saw significant differences across the board. For my root weight, not so much, and this data was supported by my ANOVA results as well. Although there are some outliers and I saw significant values, it wasn't really enough to conclude that there is an overall significant difference in the root that is caused by an increasing environmental abundance of silicon dioxide. For the total weight, I did see a lot of significant difference, and then for the total weight, I did between just two of the groups that I conducted. All right, and from here on out, we are moving to the chemical testing portion of my experiment. The first factor that I'm testing for is root vigor, which is essentially just a measure of the amount of root growth. So a higher root vigor indicates the normal health and development of my plants and seeds, and the lower root vigor would mean my seeds are not quite as viable. The chemical that I'm using to test root vigor is triphenyltetrazoleum formazan, which is quite a mouthful, so I'm just going to refer to it by TTF. TTF is a chemical that indicates a functional respiratory chain in plants that will be the electron transport chain. This chemical is colorless when it's oxidized and it turns pink when it's reduced. And by measuring the optical density of my solution, I can determine the amount of TTF that is present. So right up here, you can see that in my control group before and after TTF extraction, in the before it was quite colorless, I didn't really see any pink substances, and after I had extracted that solution turned quite pink. Again, I don't have solid results for these tests moving on here and out. I'm still working on this part of my experiment, but I do have some preliminary results to show you guys. So far, my preliminary data did not show me a significant difference between these groups. However, I only did that with one biological replicate just to run through the procedure, so I'm hoping to see that change as I conduct further trials. And then the last four factors that I'm testing for have to do with reactive oxygen species and oxidative stress, which is the main point of the study. So reactive oxygen species are any oxygen byproducts that are created through metabolism. They're unstable species that will readily react with any cellular metabolite products, and they contain one or more free radicals. A buildup of reactive oxygen species is the main mechanism of many pathophysiologies. Reactive oxygen species can cause DNA damage, RNA damage, they can cause proteins to misfold and cause them to be non-functional, and a high enough amount will cause cells to undergo apoptosis. And an increase of ROS is what directly causes oxidative stress to build up in cells. Oxidative stress, simply put, is just a difference between the antioxidants and the amount of free radicals in a cell. If the cells are unable to detoxify these products, they will undergo cell and tissue damage, and again, they will undergo apoptosis. So the first thing I'm going to be testing for is hydrogen peroxide accumulation. Hydrogen peroxide is commonly known to be an indicator of the increase in the transcription of defense and resistance genes when a cell is faced with environmental stressors. In this case, that would be silicon dioxide. The solution I'm using to test for hydrogen peroxide is DAB, because DAB is oxidized by hydrogen peroxide and it causes a visual change. It causes a dark brown precipitate to form. So in my leaf right over there, again, it's kind of hard to see up on this projector, but if you see those kind of darker brown waves running down the middle of the leaf, that would be an area where I have a higher amount of hydrogen peroxide present, whereas lighter amounts where there won't be as much hydrogen peroxide present. The next thing that I'm testing for is malondialdehyde analysis, also known as MDA. MDA is the end product of the oxidative decomposition of polyunsaturated acids, specifically the ones that are initiated by radicals, commonly known as reactive oxygen species. So MDA is a common known marker of oxidative stress and is a significant indicator of lipid peroxidation. So when I conduct this study, if I see a higher amount of MDA, that is an indicator that my cells might be undergoing some sort of cell membrane damage. They might be suffering some damage to their endogenous lipid sources. And the last two factors that I'm testing for are the SOD and the POD activity. These are both families of dismutases, which serve as antioxidant defense enzymes, and they combat the buildup of oxidative stress in these cells. So an increased enzyme activity would indicate an increased fraction of oxygen species presence in my cells. So for my preliminary data, I did not see a significant value for my SOD levels, but I did see a significant difference in the POD levels. So far from my studies, I can conclude that camelina sativa significantly impacts silicon dioxide significantly impacts the number of leaves, longest leaf length, shoot weight, longest root length, total plant length, and total plant weight with increasing silicon dioxide abundance. Silicon dioxide has greater impacts on the camelina sativa shoots compared to the roots, and further data through my chemical testing will prove the actual mechanisms through which these damages occur at a cellular level. Peers in Dr. Zhang's lab are conducting similar experiments, although they're using variable nanoparticles and they're using variable plant species. So once those studies are completed, we can do a cross-study analysis to determine if these differences are present across the board and if these differences are analogous to one another, or if this is a singular case study that is only present with camelina sativa and silicon dioxide. In the future, a CRISPR gene editing study could be conducted to knock out the genes which are impacted by silicon dioxide, and they could be used to create crop lines that are resistant to increasing nanoparticles, which could help on the agriculture industry. That's all I have. Does anyone have questions?